There I am. Okay, so today I'm going to be going through exam 2, just going through question by question. I'm going to go in order so you can skip around. There were multiple versions, so you might need to skip around a little bit more, and maybe the wording was changed, but the idea behind all the questions is the same. I'm only going to be going through one version, but if you have any issues like that, just shoot me an email and let me know. Some of us did very, very well on this exam, and some of us didn't do as well as we would have hoped. So I'm going to be trying to figure out some ways to help us compensate for maybe where we didn't do quite as well on exam 2, but we'll talk about those moving forward. So let's just go ahead and go through exam 2. Okay, what is the equation for photosynthesis? The equation for photosynthesis, well, what are we doing in photosynthesis? We're making glucose, right? So glucose needs to be on the right side of our equation here, so we can eliminate this answer. This is not the right answer. Let's see. Also, water is involved. Remember, we need to have water there so that we can replace that electron that we split off during the light dependent reactions. It's almost the right answer, but not quite. This is the most complete answer. Carbon dioxide plus water plus light yields glucose and oxygen. What happens in the thylakoid of a cell? Ooh, this is a mean question. Yeah, I apologize. So what happens in the thylakoid of a plant cell is the light-dependent reactions. The light-independent reactions are actually taking place in the stroma. So remember, you have the chloroplast, and then inside the chloroplast, everything that's not a thylakoid is called the stroma. And then you have those granum, which are stacks of thylakoids. But yes, the answer to this question is the light-dependent reactions of photosynthesis. This is a nonsense answer. These two are somewhat plausible, but the answer is just light-dependent reactions. What is produced in the light-dependent reactions? Remember, in the light-dependent reactions, we're making the high-energy molecules that are going to power the light-independent reactions. We're making ATP and that electron character in NADPH. What is Rubisco? So in the lecture, I talked a bit about Rubisco being not super great at its job and accidentally undergoing photorespiration. But Rubisco is an enzyme that helps organic carbon. No, it's an enzyme that helps fix CO2 into organic carbon. It's one of the most miraculous enzymes around, but it still kind of sucks at what it does. Where does the mass of a tree come from? I think we had a discussion question. Anyway, it's the air. It's incredible. Plants basically breathe in carbon dioxide and use it to build these massive structures that are the tallest living things on the planet. Plants produce oxygen when they photosynthesize. Where does the oxygen come from? The oxygen comes from the splitting of water molecules. So you have that electron that gets excited from the photocenter, and then basically there's a hole left over, and it needs to put something there. And so it steals an electron from H2O, and that is where oxygen comes from. The H2 basically winds up getting removed, and you wind up with just the O left over. I mean, ATP synthesis, maybe that could be an answer. Chlorophyll is kind of a nonsense answer. Electron transport chain could be plausible, but it's not quite right either. Yeah, it's the splitting of water molecules. Where do all, or at least an overwhelming majority of living organisms get their energy from originally? Kind of wordy, but I talked about this in the lecture, and the book talks about it extensively. They get it from the sun. So there are certain organisms that can actually metabolize other things like rocks, but those are in the vast minority of living organisms. Just very few kind of cool, interesting ones. What comes from the light-independent reactions? So the light-independent reactions basically are making glucose. So remember, photosynthesis is a two-part process. This might be kind of mean because you do get some NAD+, but that's not the point of the light-independent reactions. The thing you're doing here is you're making glucose. What is the purpose of cellular respiration? To convert carbohydrates into energy. You're taking that sugar, you are breaking it down in glycolysis in the Krebs cycle and the electron transport chain, and you are making a lot of ATP, or at least as much ATP as possible from that. Convert gases into plant mass, keep oxygen flowing to the brain, to convert energy to carbohydrates, that's kind of the opposite. To produce CO2, yep, convert carbohydrates into energy. That is definitely the best answer there. Which part of cellular respiration generates the most energy? That would be the electron transport chain. Mitochondria is not in the cellular respiration. Krebs cycle gives you some, glycolysis gives you some, but the big payoff here is the ETC, the electron transport chain. What is the purpose of glycolysis? To start breaking down glucose, add electrons to high energy electron carriers, and produce some energy. To finish breaking down glucose? No. Remember, glucose gets split into pyruvate. The pyruvate then moves into the mitochondria for the Krebs cycle. To start breaking down glucose, give the electrons generated in the electron transport chain a place to go. So electrons aren't generated in the electron transport chain. So this is a trash answer. More than one of these answers is correct. No, we got it right. Where is the energy in the glucose? I should have just said, where is the energy in glucose? But where is the energy in the glucose stored? In the bonds between atoms. So within neutral subatomic particles. No, who would put energy in neutrons? That doesn't make sense. The hydrogen bonds between molecules, that's almost an okay answer, but there isn't very much hydrogen bonding happening within one atom of glucose, or one molecule of glucose rather. Within positive subatomic, within the protons, no. It's between the bonds, or it's in the bonds between atoms. That's where the energy is actually stored. Which of the following is not an electron carrier? Well, NADH is, FADH is, THQ/TH2Q. Yeah, that was a nonsense one I just made up. I think THQ used to be a video game company. I'm not sure where I got that from. Anyway. What would happen if the enzyme ATP synthase was destroyed in all of the mitochondria in your cells? The electron transport chain would be slowed significantly until other enzymes were able to compensate. No, other enzymes wouldn't be able to compensate. The electron transport chain would be able to proceed but a significantly reduced efficiency leading to eventual death. That's kind of a nonsense answer too. The electron transport chain would not be able to proceed leading to a lack of ATP and ultimately death. That's my answer so far. There would be a lack of NADH, which would lead to cell death. No. NADH are dropping off their electrons at the electron transport chain, so yeah, more than one of the answers, no. This is the answer. The electron transport chain would not be able to proceed. Remember, ATP synthase is like the final enzyme in the electron transport chain. It's going to spin around because of that proton gradient that we built up. You guys are like, what are you talking about? That was weeks ago. Anyway, electron transport chain would not be able to continue. What happens to the citric acid cycle? Pyruvate, product of glucose breakdown, is regenerated into glucose. Now, pyruvate is further broken down, generating FADH2 and NADH for the electron transport chain. So this bottom one's a garbage answer. This is what's happening in glycolysis, not during the citric acid cycle. Pyruvate regenerating the glucose, that's another garbage answer. So let's keep going. You are observing an organism with 25 pairs of chromosomes, so 50 total. go through meiosis. How many chromosomes will the resulting cell have? So it starts out with 50 total, and you need to have that number, so we're going to wind up with 25 total at the end. That's what we're doing in meiosis. We're making haploid gametes from a diploid cell. We wind up with 25 total chromosomes. You are observing an organism with that same stuff we just read. Go through mitosis. How many chromosomes will the resulting cell have? The exact same number that we started with, because in mitosis we're generating identical cells to what we started with. That's the whole point of mitosis, so that you can grow, so that you can repair yourself. So you need to have the same number at the end as what you started with. Okay, the below image is undergoing mitosis. Which phase of mitosis is this in? So you see the chromosomes here have condensed, the nuclear envelope is breaking down, the centrosomes have migrated. This is just kind of a you know it or you don't. I'm calling that prophase. The chromosomes haven't yet lined up on the metaphase plate. There's still some nuclear envelope. We're calling it prophase. How would you describe the ploidy, haploid or diploid, of a cell undergoing meiosis that is just starting prophase 1? That cell is still diploid. The sister chromatids haven't separated yet. The homologous chromosomes haven't separated yet. This is still a diploid cell. That's a mean question, I'm realizing now. I apologize. I'm going to apply some sort of curve to this exam, but that was a mean one. Yeah, because I told you I pounded into you. Meiosis is for making haploid cells, and then I got super nitty gritty. Anyway, let's move on, but I do recognize the meanness of that. After the fact, of course. How does your DNA spend most of its time in an uncondensed state known as chromatin? Remember, it's like a bunch of yarn that's been scattered throughout a room, and then when you start undergoing prophase, you're gathering up all of that yarn into like a ball that looks like an X. But it's most of the time your DNA is in an uncondensed state that we call chromatin. Let's go back for a second, actually. So duplicated, waiting for cell duplication. This isn't true either, by the way, because your cell can enter G0 when it never duplicates its DNA, or it can be just in the stages before duplication. But your cell's not going to spend most of its time with its DNA duplicated. Homologous chromosomes can be described as two chromosomes with similar genes in similar locations that you get from each parent. You get one from your mom, you get one from your dad. Those are your homologous chromosomes. Here's my gene from my mom for being tall. Here's my gene from my dad for being short. Remember all that stuff. So that's the answer to homologous chromosomes. This one is sister chromatids. I don't know if I asked about that, but we'll see. The term diploid means having both sets of genetic information. You have each set of your homologous chromosomes. Required fertilization? No. Required fertilization? No. Having only half, this would be haploid. Both sets of genetic information is diploid. What are the identical copies of DNA that are created during synthesis phase of interphase referred to as? That's a wordy one. These are your sister chromatids. Sister homologs don't exist. Homologous chromosomes are the different versions, and they could also be called chromosomes, but referring specifically to those two identical copies. So they are sister chromatids. What would the resulting cells most likely look like if S phase, synthesis phase, and interphase was not able to replicate DNA? The chromosomes would replicate themselves during metaphase in order to compensate for the lack of DNA. Nah, that's a nonsense answer. That's not how it works. Each cell would have a normal amount of DNA. No, not if they weren't able to replicate the DNA. Each cell would have a half of each chromosome. That's not how it works either. Your cells can't just break the chromosomes in half. That would be really bad for you. The cell would be stopped at the G2 checkpoint. Remember, we talked about all of those checkpoints that are going on. You have G1, G2, and M checkpoints. So this one's going to get stopped before it can even move on. It's got the inappropriate number of chromosomes, so it gets stopped right there. What would a cell's chromosomes look like if it was not able to proceed into prophase? So if it's not able to proceed to prophase, we are stuck in interphase. Chromosomes would be condensed, nuclear envelope would be intact. Chromosomes would not be condensed, the nuclear envelope would not be broken down. That sounds like the hallmarks of prophase are chromosomes condensing and the nuclear envelope breaking down. That's my answer. Chromosomes would be condensed, nuclear envelope would be broken down. No. This is so wordy and annoying. I'm going to try not to put stuff like this on future exams because it sucks to think about. Chromosomes would not be condensed. The nuclear envelope would be broken down. Centrosomes would... No. I think we got the right answer. Or maybe I'm just sick of reading it, so I just picked that one. We'll find out. In which stage is the image below? Well, you can see that you have your homologous chromosomes. You can see they've got some independent assortment activity going on where the different colors are lining up in different places. By the way, chromosomes don't actually have colors like this, but it's good for demonstrating maternal and paternal pairs. But this looks like metaphase. And since we have both homologous chromosomes, I'm going to call this metaphase 1. During which phase does a typical skin cell, for instance, spend a majority of its time? Cells spend most of their time hanging out in interphase, waiting to undergo mitosis. Remember, the meiosis part of the cell cycle is just a very small part of that big circle. Define cancer. When cells have an out-of-control cell cycle, a number of cells cluster together to form a tumor. Cancer doesn't have to be this, but it certainly can take that form. A region where cells can't replicate themselves. That would not be cancer. It would be the opposite of cancer. It varies depending on what type of organism is being discussed. No, it is when cells have an out-of-control cell cycle. What could happen if a cell didn't have any functioning cell cycle checkpoints? Well, this kind of goes back to the last question, and probably shouldn't have asked this because it's not different enough from that last question. But the cell could become cancerous. Where were we? So if the cell didn't have any cell cycle checkpoints, remember the point of those is to stop the cell if the cell isn't ready or if there's something wrong with the DNA. And so if there are those problems and the cell cycle just kind of keeps on churning and churning and churning and churning with nothing to stop it, you can wind up with a bunch of cells with really messed up DNA that are indeed cancerous. So the cell could become cancerous. It doesn't necessarily mean that it will, but it probably will. The gene X55 has a role in regulating the cell cycle. Its job is to stop cells from having an out-of-control cell cycle. What is X55 referred to as? X55 is a tumor suppressor gene. Remember, a tumor suppressor gene stops tumors from forming. It stops cells from having that out-of-control cell cycle. Tumor enhancer gene? No. Cell cycle protease? No. Proto-oncogene? No. And I did mess that one up in the lecture, but I definitely clarified for you the difference between onco, proto-onco, and tumor suppressor. So there it is, tumor suppressor gene. Your body is trying to create gametes so that you can reproduce sexually. Which of the following processes should it use to accomplish this? Meiosis. Guess it's not super fair that I put meiosis 2 and meiosis 1. But the best answer to this question is definitely meiosis. Meiosis 1 is not going to get you all the way there. Meiosis 2 is not going to get you all the way there. You create gametes with meiosis. You fall down and scrape your knees. Your body is attempting to repair the skin that's been damaged. Which process should you use to do this? Mitosis. Growth and repair. There it is. What is crossing over? When two homologous chromosomes exchange genetic information, when two sister chromatids exchange genetic information. Sister chromatids never exchange genetic information. Need more information to answer? No, we don't. When two non-homologous chromosomes exchange genetic information, no, that would be weird. I feel like the gene for eyes is switching place with the gene for being tall. You just get some kind of very odd organisms. So it's when two homologous chromosomes exchange genetic information. According to my lecture, when does crossing over happen? I said it was prophase 1. Technically, it's pro-metaphase 1, or between prophase 1 and metaphase 1, but I said it was prophase 1, so prophase 1 is the correct answer. That's all right. What does independent assortment ultimately result in? So what is independent assortment? Remember, it's when those homologous chromosomes can line up on either side, and so at the end you can wind up with a gamete that has a bunch of mom's genes or a bunch of dad's genes or a bunch of mom and dad's genes all mixed together. That's what independent assortment is up to, increasing genetic diversity. How are anaphase 1 and anaphase 2 meiosis different from one another? Anaphase 1, homologous chromosomes separate. Anaphase 2, sister chromatids separate. Yeah, that's the same. That's the best answer there. What is a fertilized egg referred to as? It is a zygote, and it is diploid. It has both sets. That's what happened after fertilization. So triploid can go. Embryo, yeah, kind of a mean question, actually. It's not child. It's not yet an embryo. It's not haploid, so you can at least narrow this thing down. Yeah, that was a mean one too. Sorry. Where is the genomic DNA of a eukaryote? It is inside the nucleus. Leave it at that. When considering a recessive trait, you cross two individuals who are heterozygous for that trait. What proportion of their offspring would you expect to express that trait phenotypically? So let's see. So let's make our little Punnett square. We have two individuals who are heterozygous. So put big A here, put little a here, put big A here, little a here. Here's mom and dad. Make ourselves a nice, horrible Punnett square. Yikes. So we've got big A, big A, big A, little a, big A, little a, and we've got little a, little a. So which of these individuals are going to have this trait or express it phenotypically? Just these ones. That's the only way to get a recessive trait. Little a, and remember, this is one of the four possible offspring they can have. So the answer is 1 out of 4. A group of pea plants has purple and yellow flowers. What would you call these two categories of colors? You would call those phenotypes. So if I told you that a group of pea plants has an allele that gives it purple flowers and an allele that gives it yellow flowers, then you'd call them genotypes. If I said this plant has two different positions or two different sequences that can result in these different colors, then you would call them alleles. But the answer here is phenotypes. It's not genes, it's not alleles, it's not genotypes. The answer is just phenotypes, just the characteristic that can be observed. Group of pea plants has purple and yellow flowers. One particular plant has DNA on two homologous chromosomes that cause it to have purple flowers. What would you call these two segments of DNA relative to the plant? Well, it's its genotype. The traits would be the color, which is similar to phenotype. It's not the plant's protein, although the plant is making protein with that gene, but it is the plant's genotype. It is the literal genes that the plant has. while studying a group of flowers, you see that there are several possible phenotypes for a flower, red, white, and pink. All of these traits are based on the same gene. Based on what you know about Mendelian genetics, what is the most likely pattern of inheritance? So unlikely dominance isn't a thing. Complete dominance isn't the right one here. Codominance would be both traits being expressed in equal proportion, but this is incomplete dominance because the red and white are sharing to make the pink plant. So that's an incomplete dominance there. Okay. So what kind of inheritance is shown? You have a white mouse and a black mouse. This is the same question as before. Unfortunately, I should have probably made a codominance one, but that's okay. This is, again, incomplete dominance. Maybe some versions of the test did get a codominance. If you got that, email me. According to the pedigree below, which labeled individuals do not have the condition? A and C. B and D do have the condition. That's why they're colored in. So the answer here is A and C. What is the genotype of individual C's siblings? And how can we figure that out? So again, this individual has the condition. They've got two small letters. Here, let's do this. So individual B's genotype, I don't know, we'll just use the letter B, but it doesn't mean that it's because it's individual B, but we'll just use it. But we know for sure that they are little b, little b. And we know that for sure because they have the condition. So A, what is their genotype? Well, we know that they have to have at least one big B. Okay? And we don't know what their other allele is, but let's just assume it's a little b. Okay? So let's make all of the possible genotypes for individual C's siblings. You can have a heterozygous individual here. You have another heterozygous individual here. You can have a homozygous recessive individual. You have another homozygous recessive individual. Now, are any of individual C's siblings homozygous recessive, and how do you know? They're not, because they don't have the condition. They can't be, again, because they don't have the condition. So heterozygous is the only answer to this question, and I do apologize for some confusion on these terms. I think legitimately I didn't teach this well enough, but some folks thought that maybe you could be like heterozygous dominant, heterozygous dominant, and you can't. There's heterozygous, meaning one copy of each. There's homozygous recessive, and there's homozygous dominant. So they are heterozygous for sure. Absolute certainty. They discovered that, so somebody got their personalized genomics thing. They discovered that they have a gene that would result in flowing black hair, but they are bald due to genetics. That's an irony for sure, but actually the biology term is epistasis. It's when one gene makes it so that it doesn't matter what the other genes say. So it doesn't matter if you got a gene for black hair. If you're bald, you can't tell. That gene's not being expressed. I would call that epistasis. The central dogma of biology is that DNA makes mRNA, and that mRNA is the instructions for protein, so that's not the answer. Genetic dominance, also not a good answer. None of the answers are correct. It's nonsense. So yeah, epistasis. Why are there more colorblind males than females? So remember, we males have that puny Y chromosome, and females are protected by the genes that are on their second X chromosome. So even if a female, so colorblindness is a genetically recessive condition, and so if a female has one small, I don't know what letter you want to use, one small H, she can also have a big H that will protect her from expressing that. Whereas males, if they have one small H, that's all they got. So they have nothing to compensate for it. So because females are protected by the genes on their second X chromosome. What would happen if the enzyme helicase were destroyed before DNA replication? So remember, helicase is unzipping. It's breaking the hydrogen bonds between those base pairs. And so DNA would remain bound. Base pairs would remain bound to one another, preventing replication. There wouldn't be anything to unzip it. That is my answer. True or false? DNA and RNA identical to one another. False. They are not. I talked about that quite a few times. DNA actually is missing a hydroxyl group. RNA has uracil instead of thymine. RNA is single-stranded. DNA is double-stranded. Which of the following is an accurate description of the two strand of DNA's orientation? Unimportant. Monohelical. RNA-like. Directionless. So even if you didn't know this term, you probably should be able to knock out some of these other ones. Directionless, no. Why would you even ask about that? Monohelical, two strands of DNA are double helical. Unimportant would be a hilarious but not great answer. RNA-like is maybe even the only one that's plausible. Maybe just these three. But yeah, when you're working through these, you can certainly start to eliminate some bad answers, but they're anti-parallel. They line up sort of like that. Which of the following is true DNA polymerase? It doesn't know where to start. It builds a new DNA molecule by adding base pairs, and it can only work one direction. Oh. Yeah, it can work in any direction. No, it can only work in one direction. That is my answer to that. Blank is used to connect discontinuous segments of DNA by bonding together deoxyribose sugar and a phosphate. Yeah, this is a way too wordy way of asking this question, but let's see what we can eliminate. Polymerase, no. Polymerase builds a new strand, right? Telomerase, no, but I showed you a video about what telomerase does. Helicase, we just talked about. So the answer here is ligase. It's ligating those segments back together. What would be the most immediate result of a cell that was unable to go transcription? So remember, transcription is taking your DNA, unzipping it, and making an RNA, an mRNA transcript, an mRNA copy. You don't want to take the reference book out of the library. You want to copy the information down onto your notepad and then take it out into the world and build protein with it. So no transcripts would be made, so no protein would be made. DNA would go unrepaired, no. mRNA would go unconverted into transcripts. No, that happens later on. No transcripts would be made, so no protein would be made. What would happen if a cell was unable to create mRNA? Protein would be created incorrectly? Not necessarily. Nothing. The cell would just enter G0? No. After existing mRNA was degraded, no protein would be created. Yes, we wouldn't be able to undergo transcription, and so translation would also, by necessity, come to a halt. What is the function of tRNA? Matching the correct amino acid to specific instructions from the mRNA. Matching the correct amino acid to specific instructions from mRNA. Matching the correct protein to the correct DNA transcript? No. Matching the correct anticodon position of mRNA with a particular DNA segment? Absolutely not. Matching the correct amino acid to specific? No. That is the answer. Okay, so let's see. We're making a segment of mRNA, right? So you should remember that mRNA, I made this easy. mRNA doesn't have any U's. And so it's either need more information to answer, which you don't, or this. So T's get converted into A's get converted not into T's. If it was two complementary strands of DNA, A's would get converted into T's, but A's are turning into U's, C's to G's, G's to C's, T's to A's, A's to U's, C's to G's, G's to C's, and so on. So that is my answer for that. What is translation? You're taking your mRNA and you're turning it into protein. The process of making an mRNA transcript from DNA is transcription, We're in the second half of that translation. What would happen to a cell if all of the tRNA was destroyed? mRNA would build up inside the nucleus? No. Translation would continue, but translation would halt. Transcription would continue, but translation would halt. I like this answer. You can still make mRNA transcripts, but you just can't convert them into DNA without your tRNA. mRNA would build up inside the cytoplasm, boosting the cell's metabolism. Funny answer, but not the right one. What is transgenic biotechnology? Well, it is taking the genes of one organism and putting it into another. That's what makes you transgenic. So if you take the gene for HGH or human insulin, you stick it into a bacteria or a yeast as a transgenic organism. That is what transgenic biotechnology is all about. What are restriction enzymes? Those are those little scissors. Proteins made by bacteria that can cut DNA at specific locations. I'm going to ramble for a second, but those were originally a bacterial defense system against viruses. Ramble was shorter than I thought it was going to be. Good for me. What is a plasmid? A segment of chromosomal DNA? No. A portion of human chromosome? No. A segment of linear DNA that cannot be replicated? No. It's a segment of circular DNA that can be used to add genes to bacteria. It's not just bacteria. You can also use them in yeast and certain other eukaryotes, but it's a little piece of DNA that you can use to add to bacteria. They do use them in nature. It's how they exchange some genetic information. That is a plasmid. Is it possible to create human proteins with bacteria? Oh, goodness, yes. No, it's never possible. That's wrong. Generally, no. No, that's wrong. Yes, but only if it's a protein that shares exact homology between the two. No. Bacteria don't grow human insulin. They don't grow human growth hormone. They don't grow MSG naturally. There is no homology for many of the things. If we were actually in lab this semester, we were going to take bacteria and cause them to glow under a black light. Okay, there's no homologue for that within bacteria, which you wouldn't know, but it is possible to grow human proteins within bacteria. True or false? The wealthy can now have the ability to clone their pets. Absolutely true. If you checked out my lecture on Dolly, you would know that it is very possible to clone higher eukaryotic organisms, and people are paying like $100,000 or like $50,000 to get their pet cloned, which is kind of weird. Kind of cool, I guess, but kind of weird. Happy Halloween. Click here. Cool. Submit quiz. We'll see how we did. I probably missed one or something. Does it grade it for me? Hey, I got 100 points. So, yeah, that was exam 2. If you have any questions about stuff, if you want to argue with me about one of the questions or answers, that's totally fine. I'm going to be working on figuring out how I can curve this in a fair way, and look out for more assignments this week. All right, cool. I will see you guys online.